Okay, so this is the update on the latest improvements to MiSeq. So this slide is really an update on the MiSeq. Many of you will have a MiSeq or be very familiar with it. So I just wanted a quick update on the instrument since launch in September 2011. So we've placed over a third of our instruments in clinical or translational labs, and many of the applications that they're used for are listed here, such as amplicon sequencing or QCing large-scale runs, sequencing bacterial genomes, infectious disease, and in a particularly interesting application, counting affected alleles in fetal blood. So this is the performance of the MiSeq as it is today. So you can achieve 1.5 to 2 gigabases of yield from a read length of paired 150 base reads using 5 million clusters passing filters, and this takes just over a day. And the performance update that we are introducing in 2012 will allow a MiSeq to generate 7 gigabases of data from paired 250 base reads. The number of reads has gone up to 15 million, and the runtime for that same paired 150 base read is now just a little under 24 hours, with the same quality as you saw with the original MiSeq. And the way that we've done this is by improving the chemistry through improving the sequencing polymerase, allowing much faster kinetics. So our chemistry cycle time has come down from 3.5 minutes to now approximately 2. And we've also got some novel reagent formulations as well for the MiSeq platform, allowing us to run much faster. And also increasing the imaging area and accessing the second surface on the flow cell. So this shows some data from one of our paired 250 base runs. So the yield in this case was 8 gigabases of data from 16 million clusters passing filters. A large number of those were perfect, as shown here, and the error rates are really very nice, certainly for paired hundreds, below 0.2% on both read 1 and read 2, and also very nice error rates for out to 250 cycles on both read 1 and read 2. And as you can see in the bottom panel here, a very high fraction of high quality bases. So in the overall run, 83% of those bases were greater than or equal to Q30, with 4.7 gigabases of perfect reads. We've also been extending read length out further than 250, and on this slide shows our latest update to push read length out to paired 400 base reads. So the yield in this experiment was 3.4 gigabases of data from 3.8 million clusters. This wasn't a full-scale run in this case, just a subset of the tiles on the instrument. One of the things that we found is that we were seeing a very high data quality again. So on read 1, over 80% of the data was greater than or equal to Q30 with the single 400 base read, and over both reads, over 70% of the data greater than Q30. So we still have work to do on this protocol, and we're looking to further improve the error rates on both read 1 and read 2 before delivering this as a product, which is what we would like to do as soon as possible. But just to show you the latest update, the longest perfect read that we've achieved so far is 678 bases. This was achieved with a 500 base human library, and we stitched together a read 1 and read 2, looking for perfect overlaps, and found the longest read in this particular library of 678 bases. So this wasn't an optimal library for this particular demonstration when we're looking to extend the read length further. I'm very confident that we'll be able to do that.